Oh, hello everyone. For today, I'm going to explain about the introductions on slides for practice in histopathology, particularly slides from the gastrointestinal changes. There are in total 3 slides for the gastrointestinal organ histopathology. First slide is about the coccidiosis in chicken. Next, there is also coccidiosis in cattle, and last but not least is the proventriculitis in chicken. So in front of you now is the slides for coccidiosis in chicken. So the tissue of the slides came from the cecum of chicken, and from here you can see that the chicken. From here you can see this is the intestinal mucosa here, and this part here is the area that we call as cecal core. This is actually the exudates of the coccidiosis inflammations in the cecum. We call it as typhlitis. So this kind of typhlitis also produce exudates and inflammatory exudates. This one is the inflammatory exudates, and this one is the intestine or the cecum itself. So I'm going to zoom this part first. We're going to zoom the part of the exudates. Okay, I'm zoom it here. The reddish materials that you can see here, this one here, this is actually a fibrin. So I'm going to increase the magnifications. The reddish part here, this is the fibrin, and then you can see some small nuclei here. This is actually comprised of necrotic tissue. So fibrin, necrotic tissue here. So if we look with higher magnification, the necrotic tissue also composed of inflammatory cells, lymphocytes, macrophage, neutrophils, and so on. You can find here. Because this exudates contains lots of fibrin, this kind eosinophilic materials or reddish material here is fibrin, admixed with necrotic tissue, so we call it as fibrinonecrotic exudates. So you will find the exudates in the lumen attached with the mucosa. Next, let's see the intestine tissue. Okay, so this is the cecum, large intestine. From here you can see there are a short villi. That's one of the characteristics of cecum. Cecum has short villi, short villus like this, and you can focus in the mucosa, in particular the epithelial and the crypt parts of the intestine. Okay, I'm going to magnify this area here. So this part here actually should be covered with epithelia, but can you see here? You cannot find the epithelia because they're what we call as desquamation or sloughing of the epithelia. Here, no epithelia at all. And then this part here, we call this as crypts. Crypts here, crypts. And you will find inflammation here. Lots of macrophages you can find here, along with some lymphocytes. Here, this is the normal crypts. This is the normal crypts. But you can see also the crypts, in particular the lumen of the crypts, contains around structure like this. This is actually the oocyst of the coccidia. You can find inside the lumen of the crypts, and also here inside the lumen of the crypts, and also the crypts also undergo necrosis like this. This should be the normal crypts, but in here you can find a pycnotic nuclei or small nuclei, showing a small nuclei with bluish appearance, along with also oocyst in the lumen of the crypts. Then you can also see in the epithelia of the crypts there are also goblet cells, lots of large goblet cells. This is characterizing the formations of goblet cell proliferations. If you look further, the necrotic area can be found here. Here, the necrotic area, fibrinous materials along with the desquamations of the epithelia. And then, if I enlarge in this, in the crypts itself, okay, in the crypt itself, you will find the oocyst, and also inside the epithelia of the crypts, you can find lots of stadium of coccidia, lots of stadia of coccidial growth. For examples like the macrogametocytes, microgametocytes, you can find here. Here, even all the crypts already replaced by many stadia of coccidial growth, along with some clear cells with clear cytoplasms, indicating lots of goblet cells. Goblet cells producing mucus. Okay. Yep, I think that's the main findings in this slide. And between the crypts, you can find also inflammatory cells. Lots of inflammatory cells here, heterophils, macrophages, lymphocytes, and so on. Okay, it's easy. For the slides, you can draw the findings, and then you can submit the drawings to teachers for approval. I think that's all for the first slides. I'm going to continue with the second slides about the coccidiosis in cattle. Alright, this is the second slide. This is a coccidiosis in cattle. The findings is similar with coccidiosis in chicken. However, the shape of the coccidia stadia of growth is a bit different. Okay, so here, this is the large intestine. This is just like I said, the fibrinonecrotic materials inside the lumen. Okay, I will magnify here. This is the, okay, let's see, this is the epithelial desquamations with necrosis in the upper part of the mucosa of the large intestine. This is also, I guess, in the large intestine, in the colon. Previously in the cecum in chicken, but in cattle this is in the colon. Similar findings if we magnify lots of inflammatory cells. This is the crypts with lots of goblet cells, inflammatory cells between the crypts. You can find eosinophils, macrophages, and so on. Easy to recognize here. Crypt necrosis also evidence. Can you see here? This is actually crypts with small nuclei here, indicating the necrosis or pycnosis of the crypts. Goblet cells also can be found in some crypts, characterizing increased production of mucus. And if you look further, you can find also this kind of stadiums of coccidial growth, maybe macrogametocytes. Okay, you can also focus in area like this. If you see inside the cells of the crypts, you can find lots of stadium of the coccidia. Okay, but if you compare with the coccidia in the chicken, it's a bit different in cattle. They have more eosinophilic or more reddish color. Okay, probably this is kind of a gametocytes inside the or maybe also schizon inside the epithelial cells of the crypts. Okay, here, just like this one, easy to recognize. You can draw and then you can submit the pictures to the teacher. Okay, not so different, but the difference is only in the shape of the coccidia, and also easy to find. I recommended you to look at the small magnification 10 times for examples, or 20 times before you focus with the 40 times. I think that's all for the coccidiosis in cattle. I'm going to continue with the third slides. Alright, for the third slides is about the proventriculitis. This is the inflammation in the proventriculus of chicken. As you know that proventriculus is glandular stomach of chicken. Let's see about the morphology of proventriculus. So this is the proventriculus. So they have, from here to here is the mucosa, and this is the muscular layer, and this is the serosa or adventitial layer. It's not so difficult to find the findings here. Proventriculitis means inflammation of the proventriculus. You will see the changes mainly in the mucosa. Okay, I will increase the magnifications. Let's see with higher magnification here. This is the mucosa of the proventriculus. Can you see this part here? This is all the mucus produced by the proventriculus. So if I magnify, can you see the goblet cells? Lots of goblet cells in the proventricular cells, and then the proventriculitis itself characterized by the inflammatory cells in the submucosa, under the epithelia of the proventriculus, just like this one here. You will find numerous inflammatory cells like macrophages and mainly also lymphocytes. Here the inflammatory cells here, and then you will find also some of the glandular part. So let me show you. This part is the mucosa, and this is the glands. This is the glands of the proventriculus. This is kind of normal glands, but if you look at another sides, the glands also has infiltrations of inflammatory cells characterizing the proventriculitis. So the inflammatory cells will be seen just under the epithelia and also between the glandular parts of proventriculus. see here. Here, this is the inflammatory cells just between the glands of the proventriculus. Okay. Look at the type of the inflammatory cells, put it on your reports, and determine which inflammatory cells become the predominant cells in the inflammatory parts of proventriculus. Okay. Not so difficult, right? Oh yeah. And this is the normal proventriculus, and this part is the proventriculus with necrosis. That's why you cannot see the intact nuclei, and also the nuclei would be very small and showing purple color, indicating what we call as pycnosis. Right, I think that's all for the proventriculus. I think you can find the findings easily. Oh, by the way, this diverticulitis, we obtained these cases from broiler chicken. The broiler chicken which had a what we call as ranting stunting syndrome. The broiler chicken is really small, and as you know that the proventriculus part of the stomach, actually they produce the enzyme pepsin and also hydrochloric acid that useful for digestions of proteins. So proventriculitis may affect the protein digestions that leads to low absorptions of proteins from the intestine, from the gastrointestinal tracts, that leads to malnutrition that eventually causing the ranting stunting syndrome. That's all for the description of the slides. Good luck to you all, and hopefully you can find all the findings easily. Thank you.